The COVID-19 and the Genomic Consortium. A variant of high consequence has clear evidence that prevention measures or medical countermeasures have significantly reduced effectiveness relative to previously circulating variants. Possible attributes of a variant of high consequence is, in addition to the possible attributes of a variant of concern, the impact on medical countermeasures include a demonstrated failure of diagnostics, the evidence to suggest a significant reduction in vaccine effectiveness, a disproportionately high number of vaccine breakthrough cases, or very low vaccine-induced protection against severe disease, a significantly reduced susceptibility to multiple emergency use authorization, EUA, or approved therapeutics, more severe clinical disease, and increased hospitalizations. A variant of high consequence would require notification to WHO under the international health regulations, reporting to CDC, an announcement of strategies to prevent or contain transmission, and recommendations to update treatments and vaccines. Currently, there are no SARS-CoV-2 variants that rise to the level of high consequence. Treatment considerations for healthcare providers. Substitutions of concern for SARS-CoV-2 monoclonal antibody therapies. In the United States, there are 3 anti-SARS-CoV-2 monoclonal antibody treatments with FDA emergency use authorization for the treatment of COVID-19, bamlanivimab and etesevimab. The National Genomic Surveillance Program identifies new and emerging SARS-CoV-2 variants to determine implications for COVID-19 diagnostics, treatments, or vaccines authorized for use in the United States. Sequences with similar genetic changes are grouped into lineages, and multiple lineages can have the same substitution. Genomic surveillance efforts provide the capability to detect viruses that have reduced susceptibility to treatments more quickly. In laboratory studies, SARS-CoV-2 variants that contain certain substitutions in the spike protein cause a marked reduction in susceptibility to bamlanivimab and may have reduced sensitivity to etesevimab and casirivimab. The L452R substitution found on the B1.427 and B.1.429 lineages has been shown to cause a significant reduction in susceptibility to bamlanivimab and a modest decrease in susceptibility to the combination of bamlanivimab and etesevimab, although the clinical implication of this modest decrease are not known. The Advanced Molecular Detection Program. The program established the SARS-CoV-2 sequencing for public health emergency response, epidemiology, and surveillance, called SHERES, to coordinate SARS-CoV-2 sequencing. The SHERES collaboration include scientists from clinical and public health laboratories, academic institutions, and the private sector. SHERES will strengthen COVID-19 mitigation strategies. Large-scale rapid genomic sequencing will allow public health experts to monitor important changes in the virus as it continues to circulate, gain important insights to support contact tracing efforts, provide crucial information to aid in identifying diagnostic and therapeutic targets, advance public health research in the areas about transmission dynamics, host response, and evolution of the virus. The SHERES Consortium is led by the CDC's Advanced Molecular Detection, or AMD program, which over the past 6 years has invested in federal, state, and local public health laboratories to expand the use of pathogen genomics and other advanced laboratory technologies to strengthen infectious disease surveillance and outbreak responses. SHERES aims to accelerate the use of real-time pathogen sequence data and molecular epidemiology for the COVID-19 pandemic response. Organize and manage public health sequencing and response efforts across the United States. Coordinate and support sequencing at state and local public health laboratories across the country. Improve communication and knowledge sharing between U.S. laboratories. Develop consensus guidance on critical data and metadata standards. Reduce barriers to bioinformatic analysis and data sharing. Better align sequencing requirements and resource needs with different sources of funding, technology, expertise, and other means of support. The SHERES Consortium includes over 60 federal, state, county, local public health laboratories, several large regional and national clinical diagnostic corporations, and academic and nonprofit leaders in pathogen genomics, bioinformatics, and public health from across the country. The consortium aligns federal laboratories and public health agencies with international genomics efforts and engages the private sector in efforts to better understand the genomics and patterns of SARS-CoV-2 transmission across the United States. The SHERES objectives. The SHERES Consortium's 8 core objectives are to 1. Bring together a network of sequencing laboratories, bioinformatics capacity, and subject matter expertise under the umbrella of a massive and coordinated public health sequencing effort. 2. Identify and prioritize capabilities and resource needs across the network and align sources of federal, non-governmental, and private sector funding and support with areas of greatest impact and need. 3. Improve coordination of genomic sequencing between institutions and jurisdictions and to enable more resilience across the network. 4. Champion concepts of openness, standards-based analysis, and rapid data sharing throughout the United States and worldwide during the COVID-19 pandemic response. 5. Accelerate data generation and sharing, including the rapid release of high-quality viral sequence data from clinical and public health laboratories into both the National Center for Biotechnology Information and Global Initiative on Sharing All Influenza Data Repositories in near real time. 6. Provide a common forum for U.S. public, private, and academic institutions to share protocols, methods, bioinformatics tools, standards, and best practices. 7. Establish consistent data and metadata standards, including streamlined repository submission processes, sample prioritization criteria, and a framework for shared privacy-compliant unique case identifiers. 8. Align with other national sequencing and bioinformatics network and support global efforts to advance the use of standards and open data in public health. SHERES is a consortium of the U.S. public health and scientific community that includes federal agencies and laboratories, state and local public health laboratories, academic institutions, corporations, nonprofit public health and research laboratories, and international collaboration.